Good evening and welcome, colleagues and former colleagues, Vice-Chancellor, guests, and Professor Sanjeev and family. It is my great pleasure this evening, in my role as Executive Dean of the Faculty of Science, to present to you Professor Sanjeev Bhakhtar of the School of Natural Sciences, who is going to be completing our Science Week lineup. We've had Science Week events all week, completing our lineup with his inaugural professorial lecture, a very belated lecture, I should say. By way of advertising, this is one of several inaugural lectures we've had in the sciences over the last season. They are all available on the college's YouTube site. They need to be transcribed after the presenters, so it takes a little while before they appear. Sanjeev's lecture will also appear there, as will others later in the year. And I thoroughly recommend having a look at those through the college's YouTube channel because they're fascinating and entertaining, and a real sort of great testament to the research that has been conducted by the staff at Birkbeck. Moving on, though, to Sanjeev, Professor Bhakhtar. His studies led him to a PhD in the Department of Chemistry at Bose Institute, Kolkata, Jodhpur University, India. His PhD was titled Structural and Functional Analysis of High Molecular Mass Penicillin-Binding Proteins in Mycobacteria, supervised by Professor Jyoti Basu. Sanjeev was not content with one PhD. He then went on to do an MPhil, a DPhil at Oxford in the Department of Pharmacology. And that was with Professor Edith Sim on endogenous role of arylamine and acetyl transferase in Mycobacteria. And apologies there for the mispronunciations there. So Sanjeev moved from the University of Oxford to Birkbeck in 2006 as a lecturer. He was promoted to senior lecturer in 2009, reader in molecular microbiology and biochemistry in 2014, and then professor in 2019. So he would have given this lecture 5 years ago had it not been for COVID. And it's great that we're now resuming and catching up some of the people who missed out then on their inaugural lectures. So to say a little bit more about Sanjeev's background, he is a molecular microbiologist and biochemist specializing in innovative therapeutic strategies to combat antibiotic resistance and persistence in tuberculosis and other mycobacterial infections. He has authored or co-authored over 120 peer-reviewed publications in leading international journals, and his cross-disciplinary translational research is widely recognized in the field. His pioneering work in antimicrobial resistance in tuberculosis was featured in Nature Scientific Reports as one of the top 100 most read articles, and has been highlighted by the BBC Health News and also Vet Times. Professor Bhakhtar's service on the steering groups, he serves on the steering groups of the ISMB, Institute of Structural and Molecular Biology, of UCLTB, and is a committee member of the Acid Fast Club UK. He also holds a role of specialty chief editor at Frontiers of Antibiotics. He is a UK STEM ambassador, actively promoting social mobilization in science, technology, engineering, and mathematics. Does public outreach and scientific communication. His contributions include participation in the Wellcome Trust funded Researchers in Residence program, winning I'm a Scientist, Get Me Out of Here, and receiving both the Microbial Society Outreach Prize and Birkbeck Dissertation Supervisor of the Year Award. His research also on Persian shallots, surprisingly, maybe not, was featured in Channel 4's Food Unwrapped, and his laboratory was recently profiled in The Biologist, the magazine of the Royal Society of Biology. So without further ado, let me present to you Professor Sanjeev Bhakhtar, who will talk about to be or not to be, the fight against microbial resistance. Thank you.
Thank you, Rick. Distinguished guests, my lovely colleagues, collaborators, my students, past and present, and many who mentored me throughout my career. So lovely to see so many faces in the audience, and I'd like to thank those who joined online today for this very special day of mine. I think, as Rick said, that TB or not TB is a dilemma. So I think whether it is nobler, whether it is nobler to really suffer in silence, the persistent cough, the sweat of infectious diseases, outrageous infectious diseases, or to take arms against this sea of bacilli. And with the new drugs, whether we could end them. There is no dilemma in science. If we are not really tackling antimicrobial resistance now, I think we will be costing millions of people's life very shortly. So it is really a global health and economic challenge. However, when I begin this with that dilemma, and we think about whether TB or not TB, I'd like to take you back to a time when, let me just settle with my slide changing. Hopefully, oh. Okay, so if I could take you back to a time which really sit in my memory deeply and really influenced my life and career, and that is really my grandmom and her stories in the childhood. When she explained how difficult it was for her to leave East Pakistan at that time, now Bangladesh, to the West Bengal, which is in India, and like many thousands of other peoples, how they really use their resilience and to really rebuild their kind of life and family in a different country, in a slightly different culture there. So I think I also seen her suffering from leprosy, and that suffering was not just the health, but also it kind of deeply affected her dignity in life time to time. So it was quite kind of psychological. But that gave me a lot of determination and strength early on to get on to do something, which I know that will come up with a lot of challenges. Irrespective of that, whether that is economic or societal. Can you all hear from the back? Because I see that this mic is disturbing a bit. So whether that was societal or economic, I think I'm ever grateful for my parents to find a good school, give us education. We were two brothers. And then one thing never get erased from my life is the golden memories of my childhood. I remember that we used to go together, a group of boys, stealing mangoes from neighborhood. No one complained ever. Played cricket almost everywhere with all sorts of different gadgets. And then I don't know whether anyone recognizes this, if they have some background in India, is rolling tires and running after kites. So those were really an amazing time that I will not forget ever. That sees so much inside. But as I said, my parents gave their all to put me and my brother into good school. They were very strict missionary school where I think I had a set of lovely teachers. And I think I still remember my life science teacher who actually created so much of stories and fun that many of us, we kind of came up to study life science because of him creating that subject so interesting for us, unlike some other subjects, which I will not mention, was not as interesting as it was, the life science. Then, during the school time, I think I made some friends. So I would like to mention about Shubhobroto Shil. he was most probably, we were three in a group and really buddies and best friends in our school. Shubhabrata most probably was the most generous and very sensitive in his heart, who studied medicine, but then left home to serve missionaries, hospitals, and still in India, he is heading a hospital to look after people who need medical help. Rest of us, I think Surojit most probably will join very soon. He's on his way. He is here in the UK and in banking. So I thought if my research grant income shrink at some point, I most probably need to call him to ask, give me some support. And then I think this friendship, which kind of crossed the continent and developed not only just within us, but it kind of went into our families. But one thing you will notice that when I left India and came to Oxford, my mustache, that was gone. Well, I think it was just after my marriage. My wife suggested that you look better when you don't have a mustache. Okay, so on a serious note, I think my first research interest started when, again, I had a mentor, Dr. Krishna Chakraborty, in my college. She always, whenever she was teaching about mycology, she always inspired us by saying that what she is doing when she is not lecturing in the college, going to Science College in Calcutta and doing some fungal work. So she took us to a field trip, and I bought some spores from there and learned how to do this kind of culturing, distinguish between the mushroom and toadstool, and then I grew some. It was quite a plenty of. And then my mom snatched all of them, cooked a spicy curry at home, and the clinical trial was within our extended family. Everyone survived after that. So then moving on to my postgraduate studies, I would really like to, I had several mentors, but I'd like to bring my special paper mentor, Professor Nirmala Banerjee. He's a known geneticist and cell biologist. And he bring me to develop some kind of, to his lab to help me developing some aseptic tissue culture technique. And then I started learning about totipotency and how the plant tissues can really be grown into bananas. I didn't have big cameras in my childhood, so I think I kind of borrowed this picture from internet. But I know that I grew a couple of banana trees in our garden in the village. So that was the time when I started gaining some skills. But this was more serious when I had, after I finished my postgraduate degree, I had a scholarship, a national scholarship. I had a opportunity to visit Bose Institute in Calcutta. And on my first visit, I had a chance to meet Professor Parul Chakraborty. Within an hour, I think she was, and she still is, an inspirational leader in Calcutta. She influenced many and encouraged them to come into scientific research. I knew at that time that she worked with Hargobind Khurana, going to Massachusetts Institute of Technology. And then within 1 hour, some of the basic questions she asked me, it was part of an interview, which I didn't realize after that 1 hour was gone. And she said, there is one condition. You can start with your doctoral training in our Department of Chemistry if you really stop going to your teacher's training. Because when I finished my MSc, my intention was to go and become a school teacher and earn something, but she actually inspired me enough to get into research in the first place and focus on research. So her Department of Chemistry and her lab was led by three very passionate scientists, Professor Parul Chakraborty, Professor Monikuntala Kundu, and Professor Jayati Basu. I was given a project by Professor Jayati Basu, and that you heard the title, but I think the basic question was asked through that PhD project was that why beta-lactam antibiotics are ineffective against tuberculosis or leprosy. And you see, as soon as I got that project, there was a natural motivation for me to go and study a subject which will help understanding a bit more about mycobacterial diseases, including leprosy. So what did I find through that project? I think I will, this is a real summary of my findings, and that was published through a couple of research articles. I found out that unlike other bacterial penicillin binding proteins, mycobacterial penicillin binding proteins are kinetically labile to the drugs, to the antibiotics. And I found that if I swap the domain of this high molecular mass penicillin binding protein, I am able to sensitize this target to the drug. Also, I think I realized that if I cannot solubilize this protein, then there will be no scope for further structural biology investigation, which kind of starts with X-ray crystallography. So I managed to delete some of the amino acid residues from this high molecular mass penicillin binding protein, a membrane protein, and managed to solubilize that. And also creating a surrogate spheroblast, I managed to establish that the activity of this enzyme is not in the cytoplasm, but happening in the periplasm. I think the most important thing for me to really re-acknowledge Professor Stuart Cole at that time, because he sent some cosmid libraries for my PhD work, and they had an established collaboration already. Without those cosmid libraries, my PhD work wouldn't have really taken off. So I think thanks to Stuart. And I really established the location or locale of this enzyme in mycobacterial periplasm. Now, it was my supervisor who organized an international TB conference in Calcutta. And then it made a lot of difference. First of all, that was my first networking opportunities with the international leaders in the field of tuberculosis. And that was the first time I had the opportunity to meet Professor Edith Singh. Edith is in the audience today. Thank you, Edith. And she called me to a breakfast meeting before the conference started, and gave me a couple of different opportunities and options that that really led me believe that I could really come and do more independently some research in the field of mycobacteriology. So that meeting was very important, and it made a lot of changes in my life since. So I moved from Calcutta to Oxford, and I think I still remember night before that flying, because that was my first flying experience ever in my life. So I had a dream, and in my dream, I was seeing that I took a very crowded local train in Calcutta, and I was going to Oxford. Obviously, that dream ended with a lot of confusion, because obviously I could not find the route to take that train to come to Oxford. And I took British Airways to come to Oxford. And here I share a sad news, which I hear today, that there was an air crash where many people really died. So I think it was a smooth journey for me to come, but a lot of crying. And I saw that almost the entire village gathered in the airport, thinking about they're saying goodbye to me. I made a promise to my mother that this is only a short fellowship, I think a short scholarship that was given by Oxford University Innovation, or ICIS Innovation. I will come back. And I moved on to, within a couple of months, I got a Wellcome Traveling Fellowship when I already started there at Oxford. And then I was named, very kindly, I was named on Edith Singh's program grant, Wellcome Trust program grant, and I moved on to that 5 years program grant, and I started working on that enzyme, arylamine N-acetyl transferase in mycobacteria, because around that time, we didn't know that what is the endogenous role of this enzyme in the mycobacteria. So that was my endeavor. But as Rick mentioned about my second doctoral training, I think at another time when I will be in the reception, if you are still curious, because many people really wonder that why I did 2 PhDs. I have solid reasons why I wanted to do 2 PhDs, because doctoral training in India and doctoral training in the UK are very two different experiences. So I gained both, and I got the best out of both places to develop my independence and continued. So do you feel, and you will see some relevance to Birkbeck here, because during the daytime, I had to convince that I'm doing my job for Edith. So I had to come back to the lab in the evening time, because there were a lot of project students I looked after, and Edith was the head of department at that time and was the director of graduate training. So she hardly could see me because she was extremely busy, but that means she gave me so much of freedom. I thought that I should enjoy this freedom and do as much as I can in various aspects of my life. So obviously that was my lab time. Then I went to engage myself with the local community, because one thing I brought from India was my tabla, those small drums. So I used to play that. Either I'm playing music or I'm in the lab doing something. These were the two things I used to do. So then the community bought a lot of tablas, and they wanted their second generation to learn about this Indian classical music from me. So my Saturdays was well spent there. And actually, very much like in India, I had a lot more respect in that community setting, because people, I didn't have a car. So they said that we can pick you up, the teacher, and drop you back home if you're teaching our kids. So I was enjoying that side of my life. But then also, sometimes, occasionally, because Edith will remember that one day she called me to her office and said that, are you not spreading too thin? Are you not doing too many stuff? But I said, this is for your department, and I'm representing your department, and everyone believes that India can play cricket. So I played cricket, and that was a Jack Cox Trophy interdepartmental team, and I made the team made pharmacology to go into semi-final in that year. And if you look at the delivery, it was not bad, I think. All right. But I was doing research. So taking you back to that time, I published a number of different research articles, and two key ones which helped me to move from my postdoctoral research to my independent career at Birkbeck. And I revealed that arylamine and acetyltransferase is in Mycobacteria, are involved in lipid metabolism, and they're essential for Mycobacteria to survive inside the host macrophage environment. We also had a fantastic chemistry collaboration between pharmacology and chemistry research lab. So Edith's other PhD student synthesized some of those molecules, and using that chemistry biology collaboration, we revealed that those inhibitors are also demonstrating a very similar phenotype of the genetic mutant that I generated using a particular homologous recombination technique during my time. So anyway, those two papers and my research and my skills helped me to get a university lectureship at Birkbeck. I'm sure that if I apply now with that credential and I tell them that I played cricket and played tabla, I most probably will not get selected for an assistant professorship. Thank you, Sally. So then I think what India gave me at that time was the skills, because the validation of my research skill came from my PhD supervisors on Saturdays and Sundays, because we had 24/7 working pattern in Department of Chemistry at Bose Institute. They used to buy ice creams, and they used to tell me that when the automation was not there, I was really good at casting big gels and sequencing DNA and reading out A, T, T, G, G, C, C, all these. So I developed the skills. And when I came to Oxford, Oxford taught me how to take the lead and go and become an independent researcher. Then ISMB, the Institute of Structural and Molecular Biology, thank you, Franca, for coming here, an institute between UCL and Birkbeck. It was founded by Professor Gabriel Waxman, who could not come here. They gave me a right stage so that I can establish my Mycobacteria research lab at Birkbeck. So this was my first team picture, and I still remember some of them who are in the audience. For example, Dimitrios Evangelopoulos. He has become part of my life now. I mean, whether I'm at work or at his work or family, he is always there. and then many others who joined and helped me. However, I think early collaboration was key for me. So I think I started some internal collaboration. So Professor Nick Kip is in the audience. So that was my first encounter to understand whether ever I will be able to do some structural biology studies. And he welcomed me and we started supervising a PhD student together. And I could not let Edith go away from my life. So I pulled her, and then she agreed to help me supervising Dimitrios and Evangelopoulos between Oxford and Birkbeck. And then my other key collaborators, Professor Tim McHugh, who is also in the audience from UCL Center for Clinical Microbiology. And I don't know whether I introduced Simon Gibbons, Professor Simon Gibbons, as my collaborator. I mean, at that time, of course, that was collaborator. He was a key collaborator, but now he is more than my own brother. I don't have any elder brother, so he kind of emerged into become a friend rather than a collaborator. And thank you, Simon, for coming long way from Oman to UK for this inaugural. So then that was the beginning, but I had to expand my collaboration, and I always believed in interdisciplinary research. Professor Helen Hels in the audience from UCL Chemistry. Irene Nobeli is not feeling well, but online, bioinformatician. Then Professor Franz Booker from University of Graz, and then Del Bester from Birmingham. We started really formulating a team to really solve the problem of antimicrobial resistance and focusing on cell wall peptidoglycan metabolism in Mycobacteria. I realized that I needed one good PhD student to start my lab, but I was lucky enough to had two at that time. So Dimitrios Evangelopoulos and Chandrakala Basavanacharya. Chandrakala joined me from US, and I'm sure that she is online and listening. Those two really were my pillars at that time to build my research lab to tackle tuberculosis. And then naturally with that team, with good students, my early success came when I had Medical Research Council New Investigator Research Award and a collaborative award, which was led by Professor Stephen Gillespie, who is also in the audience. So that set the scene for me to build the network. Now, as I said, that ISMB, Institute of Structural and Molecular Biology, between Birkbeck and UCL, played a major significant role in my early career, mid-career, now, and my future is there, because I think the original vision was that they will support interdisciplinarity and they will build a collegial environment. And you can see there that how Gabriel Waxman founded this, then moved on to Finn Warner, who is also in the audience. So those two initiated the culture of interdisciplinarity, moved on to Professor Franca Fatrinelli, and she is the current director of this Institute of Structural and Molecular Biology. We always think about whatever we do is how do we integrate interdisciplinary approaches to solve our independent puzzle that we the question that we ask. And now here my role was mostly a photographer, and behind that screen you can see that made smile on many of our colleagues' faces, including Gabriel Waxman. I hardly seen him laughing out loud like this. So it was good, and we kind of started engaging ourselves. My direct boss, Professor Kathleen Thompson, who is also in the audience, the head of natural sciences. We had a good time, didn't we? And then I'm sure that Gabriel is not just posing. He found something in that orchid, but it was captured by my lenses. Now, Tina was very brave, and with Andrea, it seems like they're communicating with the star of that day. I just like to be behind the camera lens. And then I think what I liked most in that away day, in that retreat, was this team building activity, blindfolded, and everyone was having fun. At that time, I realized that this must be my turn now. So I went and joined, and it was a really fun day. Anyway, on a serious note, while we were building all this, I developed my interest in Mycobacteria. I developed more interest in the diversity of Mycobacteria and the mycobacterial diseases that is caused by this notorious, unique bacterial pathogen. Every microbiome that you can think about, whether that is skin, whether that is gut, whether that is lung, whether that is urogenital, everywhere mycobacterial species are present. Mycobacterial tuberculosis cause primarily a pulmonary disease. However, they can leave their primary site and get into bloodstream. When they get into bloodstream, they really can cross the blood-brain barrier and can cause brain TB. They can go into urogenital microbiome, create dysbiosis, and can cause child birth defect. So there are a lot of problems that we see is caused by this one genus, which is Mycobacterium. Now, I think one interesting fact that I'd like to highlight here, that all these mycobacterial pathogens, they can form biofilm and they can form granuloma, particularly the pulmonary TB. And the interesting fact, common fact about this formation is they're complex and dynamic. I think although this is my professorial lecture and good that is happening after 6 years, I don't want to end my career. I want to really jump into this complexity and dynamism that I see in mycobacteria in that physiological state, and to unravel that how fascinating it is to really tackle this bacteria living in various different parts of our body. So mycobacteria cause disease in both humans and animals. They're present almost everywhere in our body, as I said. And the interesting fact is mycobacteria inhabit both hosts, like human and animal hosts, and their surrounding environments. So mycobacteria is almost everywhere. Now, out of all those mycobacterial diseases, TB is the killer disease, and tuberculosis is still remaining to be the world's leading cause of death from a single infectious agent. Interesting fact is that the fact that I don't need to change for my teaching slide is that every year between 9 and 10 million people get newly infected by Mycobacterium tuberculosis, and between 1 million and 1.5 million people die from this devastating disease. Another interesting fact is 1/3 of the global population is carrying Mycobacterium tuberculosis in a dormant physiological state, and they wait for the host to really go through their senescence, aging, any other like infection, co-infection, which can affect our natural immune system. Those dormant bacilli can wake up and cause the disease. So an interesting question is that how to really understand all these different physiological stages of Mycobacterium tuberculosis. And often we understand tuberculosis or mycobacterial diseases are prevalent in Southeast Asian countries or African countries, which is true that they're prevalent. However, where we are sitting today, London is a major hub for TB infection. There are boroughs where TB infection rate is significant, and that raises concern for both home government and home ministries, as well as people who deal with internationalization of health and disease. Anyway, apart from that, antibiotic resistance added an alarming threat to this scientific challenge. So around 40,000 extensively drug-resistant TB cases arise every year from almost around the world, and that means literally there is no antibiotic to treat those extensively drug-resistant cases. Not a single antibiotic, not a combination of antibiotics, which is not acceptable, which means that we need to really address that, and we definitely don't need to live in that dilemma whether TB or not TB. We really need to find new drugs to tackle a TB infection there. So I want to bring a picture in front of you for you to imagine that how antibiotic actually works and how really they ultimately get rid of the bacteria. So there are small molecules of the drugs. So you see those cartoon D there. And then macromolecule or machineries, when one enzyme or multiple enzymes contribute to an essential function or conditionally essential function for mycobacteria to survive inside the host. Now, then the microbe is the whole organism, and the microbiome is where they're living. So in this case, see, there are nearly 300 microbes, 400 microbial cells living in the lung microbiome, whereas there are millions of microbes live in the gut microbiome. And how then antibiotics precisely work in this situation? That drug will get through the human body, they will get administered, they will get absorbed, they will get distributed, find their enemy, which is bacteria, and then bind to that macromolecule, that enzyme, which we know are essential for bacteria, the mycobacteria, to survive. And if they are effective in inhibiting that function, then the bacteria will disappear. Either their growth will be inhibited or they will die. So with that, I think we have a number of different antibiotics that we use to tackle tuberculosis. So all those in white are those antibiotics developed during the golden era of antibiotic discovery, and those in yellow, they developed later on and created a new combination to tackle drug-resistant TB infections. Most of them, they target cell wall. Either mycolic acid or arabinogalactan or membrane potential or energy metabolism, DNA RNA synthesis. And however, there is no antibiotic that was designed or discovered or developed targeting cell wall peptidoglycan currently to tackle Mycobacterium tuberculosis infection. So I will again go back to that complexity of Mycobacterium tuberculosis getting into our body, and 70% of the cases, our immune system are capable of getting rid of them, which means spontaneous healing. But what happened to the rest of the 30% of the cases? There you see their host pathogen interaction between the pathogen and the innate immune system or the humoral immune system, and ultimately they negotiate, and that negotiation bring them to a situation where Mycobacterium tuberculosis will either continue to replicate and leave their primary site, go into other places, cause active infection, which means that the patient will start developing clinical symptoms in case of pulmonary TB, or most of the cases, they go into dormant physiological stages, and that dormancy is resuscitated due to malnutrition, due to senescence, due to HIV co-infection, or anything that can compromise our host immune system. So that is how you see. But most important fact for you to take on here is that they live in different physiological stages, either actively replicating or non-replicating or slowly replicating. And all those different physiological stages are interregulated and very interesting features of this phenotype of this unique bacterial pathogen, which needs to be targeted by molecules. Otherwise, we will never be able to get rid of mycobacteria who can live in those different physiological stages. So existing antibiotics that I shared are mostly targeting the actively replicating phases, whereas new antibiotics or repurposed drugs that we think of can, if we are strategically developing their target areas, they should be able to get rid of both actively replicating and non-replicating bacteria. So with this background, I'll give you 3 thematic areas of my research, and I will try to summarize that where we are and where we head towards. So theme 1 is validating novel therapeutic targets for anti-TB drug design. Theme 2 is developing methods and models in new antimicrobial drug discovery. And theme 3 is repurposing existing drugs, FDA-approved drugs, and whether we can tackle with those existing molecules to the extensively drug-resistant TB treatment. So the theme 1 starts with this idea that if one could really target the fundamental peptidoglycan layer in Mycobacterium tuberculosis, we know that peptidoglycan is covalently linked to arabinogalactan. Arabinogalactan is covalently linked to lipoarabinomannan, lipomannan, and mycolic acids. So that means if you target cell wall peptidoglycan, you automatically disrupt the entire energy-demanding cell envelope of mycobacteria. And naturally, there are so many antibiotics that are already in the market and very popular in treating bacterial infection, except mycobacterial infection. And that is where my research investigation began at Markbeck. I found out something as a gap in the literature. And I realized that the step 2 of this biochemical reaction is led by 4 enzymes, sequentially adding L-alanine, D-glutamate, mesodiaminopimetic acid, and D-ALA to the backbone of cell wall peptidoglycan. So I thought that let us focus on those enzymes involved in this very important step of cell wall peptidoglycan metabolism, and whether that can be targeted, whether we can validate that as a novel therapeutic target. Chris Daniel is a final year PhD student in my lab, and she described earlier in the Science Week that how this CRISPR-I method can be used to silence and knock down number of different genes function, and we can see the phenotype, but at the same time, we can demonstrate their essentiality or non-essentiality. So using that, we created a large library of CRISPR-I deleted mutants. And we know that 7 out of 9 genes involved in cell wall peptidoglycan metabolism are essential. They're not only essential, but they're very vulnerable, because you can see, like in the audience, if you look at who you are sitting next to, you will find out some sort of functional connectivity there. So we found that cell wall peptidoglycan metabolizing enzymes like MUR-E, F, D, and C, they are sitting with FTSs, which are known as cell division proteins. We identified that they're co-transcribing and translating, and we identified that this is where we think are clustering, or as an operon, they're expressing to perform 2 critical roles. One is in cell wall synthesis, another one in cell division processes for Mycobacterium tuberculosis. We also used a protein fragment complementation system in vivo inside bacteria. We identified this airline-like network where we found that MUC, D, E, F, although they are not interacting among themselves, but they are interacting with PKN-A and B, which are major regulators of various other proteins in Mycobacteria. And then MUC, D, E, and F are interacting with cell division, major cell division proteins in Mycobacterium tuberculosis. We also found that they're interacting with the TETR regulator and some of the other enzymes which are supplying them the substrate for their functionality. So that helped us to understand not only cell wall peptidoglycan metabolizing enzymes are essential, but also they're vulnerable for us to really think about designing or discovering molecules against them. But the step 1 was to clone, express, purify, and make sure that we have some pure protein. And that is when I had to go to Nick to ask that, okay, we now have some pure protein. Can we do some structural and functional studies? Now, in my lab, we developed a high throughput enzyme assays, which helped us later on to screen thousands of different molecules to answer whether any of the molecules that comes from natural products or synthetic libraries, whether they are able to inhibit ATP-dependent MUR-E ligases function. But with the pure protein, we also developed an analytical assay, HPLC-based assay, to demonstrate that the substrates are getting consumed and the products are forming. So that is more of an accurate detection of enzyme activity in vitro. We also used various different substrates to demonstrate that how stringent these enzymes are when we are providing them the substrates compared to whether they're relaxed about reacting with anything that we provide in vitro. And with that, it was our first PhD student, Chandrakala, who managed to crystallize ATP-dependent MUR-E ligase from Mycobacterium tuberculosis. In that structure, we found there are 3 different domains. Domain 1 is primarily involved in interacting with the dipeptide-linked N-acetylmuramic acid. Domain 2 is involved in interacting with the ATP, and domain 3 is involved in interacting with mesodiaminopimetic acid. And then we identified about 12 active site residues in that enzyme, which we know play a major role in the activity of this enzyme. So if you imagine that whether we could use that as a target for new drug design, it is important for us to then know that where a medicinal chemist could anchor their inhibitor and design a drug which not only will go and bind, but interfere or inhibit this enzyme's action. So we found out that 5 of those amino acid residues, when we mutated them, the enzyme activity gone almost to the baseline. So those amino acid residues in this enzyme was found vital. Now, with this, I think the idea was to design new molecules. However, the resolution of the structure was not good enough to start any drug design project. We serendipitously discovered, or we discovered by screening thousands of different molecules against the activity of this enzyme. And the first Muri inhibitory compound was identified from a plant product, Ocotea macrophylla. Simon would remember that we supervised Juan David Guzman, came from Colombia with future funding and some other funding which was provided by UK government. And then these aporphine alkaloids which were extracted, purified, and we found out that those are not only inhibiting or killing Mycobacterium tuberculosis, but also they are inhibiting the activity of the enzyme at a reasonable nanomolar, micromolar concentration. And we published a couple of research articles around this to establish that this aporphine alkaloids could be a promising starting point for discovering more inhibitors. And that is when I think Helen Hales came in to design some more molecules. So here you see that those thioisoquinoline molecules were designed, and we recruited 2 Wellcome Trust funded PhD students between us, and we published a paper later on with Liam Martin and her to demonstrate that some of those modified quinolines are effective against Mycobacterium tuberculosis. Then more inhibitory compounds were discovered, again from natural products, Evodia ruticarpa, and we found out that they are inhibiting the growth of Mycobacterium tuberculosis, but also they're inhibiting the enzyme. As you can see, increasing the concentration of the inhibitor, they're inhibiting the substrate consumption, the product formation there. So then Irene Novelli, a bioinformatician, helped us to dock these molecules against the enzyme structure, and we found out that these quinolone molecules are interacting with the natural dipeptide substrate, and their competition is at a uracil recognition site somewhere there. However, to convince that our molecular docking studies were valid, we performed some enzyme kinetic inhibition studies, and you can see that when we increase the concentration of mesodiamyopimelic acid or ATP, there was no change in the velocity of the reaction. However, we changed the concentration of the dipeptide, we have seen the inverse velocity of the reaction in this Michaelis-Menten Lineweaver bar plot. It kind of changed. So that confirmed that these inhibitors are competing with the dipeptide substrate binding site of the enzyme. Franz Bucher and his group went on to make a lot of other quinoline molecules, and we continued to see question number 1, whether they're more effective against Mycobacterium tuberculosis. Question number 2, whether they're more effective against the enzyme target in Mycobacteria. So to summarize that, ATP-dependent mu ligases in Mycobacteria, we know that they're essential for mycobacterial growth and survival. We know that cell wall peptidoglycan is not present in eukaryotic cells, so there is no eukaryotic homologue of mu ligases. Mu ligases are part of an operon, and they participate in major regulatory pathways. Crystal structure of ATP-dependent Muri ligase was solved and published, and I think in a couple of months' time, you will see a high-resolution Muri structure from Mycobacterium abscesses will be published as well, which will help us to really initiate a new drug design project based on cell wall peptidoglycan metabolism. The active site pocket is tractable enough for large natural products, so small molecules to get in and inhibit their function. And we developed a high throughput non-radioactive assay, which means that we are able to scrutinize, to evaluate thousands of different molecules against this valid enzyme target. And we already developed 2 hits against this enzyme, and both of them came from natural products. Theme 2, I will describe that how important it is that whatever method or model that we use in our microbiology lab in vitro, how we can pull them closer to an infection model inside the cell. And with that ambition, I think we started developing a number of different methods. And by that time, I think Simon and I started developing a more closer collaboration. We believe that nature is an amazing chemist. So let's explore all the natural product in the libraries and resources that he had in that UCL School of Pharmacy lab. And we recruited Cynthia Manning-Dankwe, who is also in the audience. And she found out that this simple pyridine N oxide molecules extracted from Persian shallots, I'm sure that View used the greenhouse and extracted several different varieties of Persian shallots. The Allium sticktatum produced these simple molecules, demonstrated a high effective healing against Mycobacterium tuberculosis. And that is when I had an early morning phone call. I was thinking that early morning call is from Sweden. It was not from Stockholm, it was BBC Breakfast Radio. And they asked me some questions. How did you discover this? What is the merit? Where are you going with these molecules? But in the end, they asked me that how much Persian shallots you think we need to eat to get rid of Mycobacterium tuberculosis. So that's when it helped, because I then remembered my math teacher in the school, and I quickly calculated and I said, roughly around 5 kilograms of Persian shallots in the morning. And imagine that you are commuting in the busy, crowded London tube. Anyway, that actually signifies that how important it is that extracting, purifying, and identifying these beautiful molecules from natural product, but it has to be synthesized in a chemistry lab so that we can take that project forward. Later on, I think Kate, who came in from Channel 4, and that was in our microbiology lab, she was very excited to know about this Persian shallot story, and mainly she wanted to know that which method that you use to discover these beautiful molecules. And this is the method. I mean, it took me 20 years. It started initially in its lab, and I tried to forget that phase because I want to bring all credits to Bart Beck about this method. But I remember that during my evening studies, I was trying out this the method. And then it kind of went through a number of different publications. Every time I published, and that was the time when I spoke with the business unit. And they said, next time, when you think about publishing, come back to us. Now, what is this method? Very simple, rapid, high throughput, and have only 4 steps, which will help you to understand whether a drug or drug-like molecule or inhibitor are killing or inhibiting growth of a microorganism. It's not just about mycobacteria, it is about any bacteria, and even we tried with some of the fungal strain. And I think we have Sudax sitting there. I went to her inaugural lecture a couple of weeks ago. Sudax brought in with that question that, can this method be used to test the antimicrobial susceptibility of fungal strain? Yes, it was possible. And then we kept modifying this. Every time a new student comes in, I said that let's create a challenge and ask about what are the disadvantages and how we can improve it. So this is the latest modified HT Spore I method. Then we optimized this, validated that. There came the Medical Research Council technology, which is now LifeArc. They gave us thousands of different molecules retrospectively to test whether that works using our method. Now, from that method, I created iSpot AMR platform, and this platform will be the new generation platform which will rapidly diagnose an antibiotic resistance in clinical isolates and will help us to detect a lot more molecules in a much faster way, but most importantly, that environment is closer to the in vivo infection model. And that is when I have to stop, because I know that there are business innovation unit colleagues sitting there, and they said, no more on this. This time, do not publish. We need to submit this to protect this work. So we submitted a patent application, and a couple of weeks ago, we submitted a UKRI proof of concept application. So if you are reviewing this application, I'm sure that you will see the merit of this new platform and how that helped in developing a new strategy to detect antimicrobial resistance and accelerate the discovery of new molecules, which we desperately need. So these are the publications which I brought as a summary, and I showed you that how we've gone on to protect this technology and trying to translate, commercialize our product for future benefit of humankind. Theme 3, and I promise you this is the last theme, which came with a serendipitous discovery. I went to a local pharmacy, bought several different non-steroidal anti-inflammatory drugs like ibuprofen, aspirin, carprofen, loxaprofen. And I found out, interestingly, because that method was already developed in Bombay, that some of those were killing killing Mycobacterium tuberculosis specifically. So the question was natural for us to ask that what is the mechanism of this antibiotic action? Because you know, ibuprofen, carprofen, they're non-steroidal anti-inflammatory. But our question is, if they're killing bacteria, that means they must have a bactericidal mechanism of action, and that is where we are heading towards. So we established the background. We realized that there are some endogenous targets which are potentially the primary target of this group of molecules. And not only they're killing Mycobacterium tuberculosis or Mycobacterium abscessus, but also they're targeting a mechanism which is called drug efflux mechanism. And that means that these molecules have the capability of reversing the tide of this antibiotic resistance. If we think about these molecules as an adjunct with the existing antibiotics. And I mentioned about biofilm at the very beginning of my talk. So I demonstrate that how these non-steroidal anti-inflammatory drugs are interfering with the biofilm phenotype. They're inhibiting the formation of biofilm in Mycobacteria. There came Professor Simon Waddell, who analyzed this transcriptomic data and identified some of those endogenous mechanisms which are a potential target for these carprofen molecules. And then we went on again using this CRISPR-I technique and created that library now for us in next 3 years with an UKRI funding to investigate that what could be the primary antibiotic target for this non-steroidal anti-inflammatory drug. Here I bring most probably the last scientific slide to demonstrate that how this iron acquisition and homeostasis are very important. And the reason for that is there is a link between non-steroidal anti-inflammatory drug and this iron acquisition and homeostasis in Mycobacteria. So all these siderophores like carboxymycobacterium, they go out, fetch ferric iron, convert them to ferrous, and then when they bring them back, there are at least 10 different iron requiring biochemical pathways that they contribute. If there is any excess, they get stored in bacterioferritin. Now, to understand, to unravel these complexities, we need to study nearly 20 different enzymes in our lab. And that is why I think we need more project students coming into my lab and come up with your idea to really see that how in a shorter time, with a limited funding from elsewhere, we can unravel this complex iron acquisition and homeostasis mechanism in Mycobacteria. I need to highlight Professor Venkatesan Jayaprakash from Birla Institute of Technology in India is one of my longest standing collaborator on this project, and Dr. Federico Bruccoli, who is in the audience from Leicester de Montfort University, and Chris Daniel and Gaurav Rakshi. Those two are amazing PhD students working on this project. So keep an eye on this project, and I'm sure that there will be some interesting publications coming up. Alongside all these biological mechanistic studies, Helen Hels and her group wanted to really explore that how we could make analogs of this carprofen and can make them more effective. So there are 3 findings that I want to highlight today. Is through her synthesis and our evaluation, we found out that these carprofen analogs are they're easier to synthesize. Remember that the carprofen industry lost over ibuprofen because carprofen synthesis is more costly and time consuming, very difficult. So that problem was solved by Professor Hell's group. The second is these analogs are more effective against Mycobacterium tuberculosis in terms of killing them. And then the third one is they're less cytotoxic against eukaryotic cell. So they bring that potential to be future antibiotic or adjunct in the chemotherapy against Mycobacterium tuberculosis. And you cannot imagine translating any of this fundamental work to the clinic until you start talking with people who then can start thinking about preclinical trial studies. So we are fortunate that we developed a long-standing collaboration with Bangalore, a group called Foundation for Neglected Diseases Research, and then Dr. Parvinder Kaur and Dr. Shandil from that FNDR, they agreed to collaborate us and they started designing some preclinical trial studies using carprofen against a number of different animal models. And we have some promising results, which we haven't published yet. So in summary, carprofen, which is like any other common non-steroidal anti-inflammatory drug, they show effective intracellular chemotherapeutic potential against Mycobacterium tuberculosis for further development. Iron deficiency potentiates carprofen anti-TB action, and that links up for us to investigate iron acquisition and homeostasis in Mycobacterium tuberculosis. There are key genes in this iron acquisition and iron homeostasis and iron acquisition is influenced by carprofen. So therefore, it is important for us to really understand whether we could identify the primary mechanism of carprofen there. And then we synthesized some novel carbazole analogs and found them to be more potent against tuberculosis. Now, obviously, I always dreamed about editing a book, which was never possible. However, until now, there were many editors who actually were convinced that I could write at least some chapters. So there are a few chapters in this microbial pathogenesis, and I'd like to thank Professor Uday Kishore for trusting on us. I'd like to thank Nicola Laurian in this team again for trusting us, me and Dimitrios, to write a chapter on that book. And then Venkatesh and Jayaprakash asked us to write a chapter on their book. And then the most favorite book of mine, not book, but book chapters of mine, is in Antibiotic Resistance Protocol, that is edited by Stephen Gillespie. And here we published two different chapters, one on highlighting how we can use CRISPR-I technique to identify the essentiality or non-essentiality of a gene, and the other one highlighted this modified spot culture growth inhibition assay, which was led by an Embrace student at Birkbeck. I do not know whether Anush is in the audience, but Anush worked on that effectively to help us to publish that. Now, obviously, by now, you realize if we go back to the TB or not TB, I think you will understand that science is not enough to tackle this global challenge. So I was always interested in reaching out to the community to tell them that how antibiotic resistance develop and how we can translate our complex understanding into simple language. So you will see some testimonial here. You need to keep an eye on the subtitles for you to understand what they're saying. This is their testimonial.
টিভি ব্যাপারেও জানতাম কিন্তু আমরা এত ভালো করে জানি গরিবদের বলে লেখাপড়া নেই সাহায্য করবে এরকম ভাব মানে তারা তো সেই বিষয়টাই জানে না সেই জন্য বাচ্চাদের মধ্যে ছড়িয়ে যাওয়াটা খুব দরকার
প্রোগ্রামটা যদি পাবলিকের সামনে ওপেন করা হয় দেখানো হয় নিশ্চয়ই তারা অনেক কিছুই জানবে এবং সচেতন হয়ে চলবে
রোগকে সবাই ভয় করো রোগীকে নয়
যেমন গান করে হলো পপেট শো দেখে হলো এরকম করে দেখালে সবাই মজাও পায় আর বুঝতেও পারে পুরো ব্যাপারটা সবাই আর আমার সব থেকে পপেট নাচটা বেশি ভালো লেগেছে
We are also collaborating with the
So I'd like to acknowledge Suresh Basu, who was a student at UCL, and she was the founder of this Joy Book initiative. And the idea was that how we can really mingle the music and the local folk art and translate our complex scientific language into a simple term so that people can understand what we are trying to tell them and explain that actually this antibiotic resistance can be tackled even going beyond the lab door. So that was the idea. However, now moving on to a time when I'm so proud of people who gave me the opportunity to mentor them, whether they are my PhD students or MSc project students or Embrace students. So I'm incredibly proud of Dimitrios Evangelopoulos, Cynthia Amanning Dankler, Jody Fellan, and Doris Key. They're all either assistant professor or associate professor in their respective universities. Two of them are in the UK, two of them are outside the UK. There are some who've gone into senior school teaching. Then we have Brian Ho and I supervise Rita Ramalhate, who is now in publishing industry, and then biopharmaceutical industry. There are some PhD students and project students who thought that that will be their place to go and develop their career. I have some PhD students, and I think with the help of Professor Tim Mackey, who is in the audience, we created a career for them, either in St. George's Hospital, Professor Phil Butcher is here in the audience, or in Royal Free Hospital. I think they developed their career as a clinical trial manager. And then a fellow came from India to Birkbeck as a UNESCO L'Oreal fellow. She's now a senior application scientist in CDG and working in the field of data management. So now my time to really thank my parents again for their incredible support. They gave all to me and my brother to make sure that we get the right education and develop our career, whether that is in the UK, because they visited us in the UK, and they were very happy for me doing whatever I'm doing in the UK, and saw that how I was really part of the British national ambition to deliver the higher education in this country. Then I'd like to thank my parents in laws, because I think they trusted me. Their daughter tied her hand with me, and I think we are going on with that. I cannot go away without acknowledging that most of the work outside Birkbeck and all the stress that one has to take is hard, so that I can deliver my teaching, my research, and etc. and etc. We met in 2020. In 2002, in 2003, we got married. But in that year, I must bring, because I was at Oxford, and I remember that I paid a huge amount of money to British Telecom, because that year was a very difficult year for me. I'm sure that many who will understand, who are at least 20 years younger than me. Then we had a pleasure to bring our daughter, the only daughter, Srija, and she was always there, as I said, like with her camera, and some of the pictures are taken by her. Now, Srija, I think because of Devashmita, that she took care of all the difficult responsibilities, so I could enjoy my parenthood. I think time to time, we've gone into various different things that we enjoyed. And I think this year, September, she will be going to study in the university. So when I joined Birkbeck, I think Srija was just born. And now that I'm giving my inaugural lecture, Srija will be going to her university. Now, the coincidence is, last week when I was in University of Surrey giving a seminar, she was sitting for her maths exam, A-level, paper 1. And today was her maths paper 2. And she said that, was that deliberate that you are not allowing me to attend any of your events? Unfortunately, I think this was just a scheduling thing. This is my last slide. And you remember that I brought this pair of tabla with me when I moved from India to the UK. And I had a 10 years period at Birkbeck when I was entrusted to be an assistant dean for internationalization and partnership. So this was our idea, to create an environment, particularly thinking about international students coming to Birkbeck, that how we could really engage them in a way that all these communities come together and celebrate in that one world festival. And I'd like to thank all those people who helped me, including it was a motivation coming from the dean at that time, Nick Gibb, suggested that, yes, it's a good idea, go ahead. But also there were many staff and colleagues who helped me to organize this. And I'd like to thank Naomi Adams, who is not at Birkbeck, but she was the linchpin of this entire organization there. So I will stop, but I would like to entertain you with about 30 seconds of that music that says, that you can go around the world in 45 minutes, but it will, I promise you, it will not be more than 45 seconds. And then I will compose a bit of my own music with that, so that you go away with some sort of musical note here before you start and celebrate this evening with wines. so much for your attention.
So many thanks again, Professor Sanjeev Bhattacharya. A wonderful combination of the personal and the scientific. Wonderful also to see the translational research, the outreach work that you've done. I think that's really incredibly important. And you know, as a colleague, I think I've known you here about that for 7 or 8 years, I guess. It's been great working with you when we're both on the School of Science faculty executive. Anyway, now we have a drinks reception outside. Traditionally we don't have questions to the speaker after the inaugural lectures, but you'll be able to speak with Sanjeev outside. And once again, congratulations.